Good morning, student. Today we are study about the ultrastructure of nucleus. That nucleus, those are mainly found in the eukaryotes, but not in the prokaryote. In the yesterday class, I already have teach it. Okay. Then I think these eukaryotic cells they having I think a nucleus. That nucleus first it will be discovered by Mr. Robert Brown. First, it was discovered by Mr. Robert Brown. Then, sir, what do you mean by nucleus, sir? Then, these are the largest cell organelle in the cell. Already you will know about the mitochondria and cell organelle, then ribosomes and cell organelle, all these things. Okay, but among all these cell organelle, this nucleus, it is what largest cell organelle. It will consist of what genetic material. Okay, it having what genetic material that is called as I think a DNA is there, or it may be RNA. Then it will control all the metabolic activity of that cell. I think within the cell you will see that I think nucleus is there. Then that cell, that nucleus, it will control all the activity of that cell. That is called as what nucleus. What do you mean by nucleus? It is what largest cell organelle, and it will consist of what genetic material. Yeah. Then it will control all the activity of the cell. Then I think sir, what is the shape of the nucleus, sir? It may be I think a lens shape, it may be spherical shape, or it may be oval shape. Okay, that one is I think shape of the cell. Then I think this nucleus, it will consist of I think 5 components are there. The nucleus I think this one is what? And cell is there. This one is what? And cell. Within this cell, you will see what? Nucleus is there. Okay. Then this nucleus, it will consist of 5 components mainly. The first one is what? Nuclear envelope. Okay. Then the second one is what? Nucleoplasm is there. Then the third one is what? Nuclear matrix is there. As well as I think fourth one is what? Chromatin is there. Then fifth one is what? Nucleus. Okay. This one is what? The structure of the nucleus. It will consist of 5 components. Then the first one is what? Nuclear envelope is there. Okay. This one is what? Nuclear envelope. Then that nuclear envelope, it is what? Completely double membrane structure. You will see here both the outer membrane is there as well as inner membrane is there. Okay. It is double membrane structure. It will surround the nucleus. Then each membrane it will measure about 70 to 80 angstrom in thickness. This one is outer is how much? 70 to 80 angstrom. Inner membrane is what? Membrane is what? 70 to 80 angstrom. This one is the thickness of the outer as well as inner membrane. Then there is a space between the outer as well as inner membrane. This one is what? Outer. This one is what? Inner. It is present in here within the nuclear envelope. This one is what? Nuclear envelope. It having the two regions, the outer membrane as well as inner membrane. In between outer as well as inner membrane, there is a space is there. Very important. That space is called perinuclear space. Which one? Perinuclear space. It is about 200 to 400 angstrom. Then it is a small cylindrical channels. Those are called nuclear pores. So within this nuclear envelope, here you will see some gaps are there. Is that what? Some gaps are there, and these gaps are called nuclear pores. We can talk about nuclear pore. Then these connect the nucleoplasm with the cytoplasm. Within the nucleus, we will see the matrix is there. Within the nucleus, you will see what? Matrix. Then that matrix is what? Nucleoplasm. Okay. Then this one is a cell. Here you will see the what? Cytoplasm is there. Then this one is nucleoplasm. This one is what? Cytoplasm. The connection between the nucleoplasm to the cytoplasm, it is connected by which one? Nuclear pore. Okay. Then I think each pore, it is not a simple pore, it is what? Very complex, very fantastic. Then it has a 120 nanometer diameter with 2 rings are there. There are 2 rings are there. One ring is present in the cytoplasm, other one is in the which one? Nucleoplasm. Here I won't draw. Here extra I won't draw. This one is what? Nuclear envelope. Yeah, this one is what? Nuclear envelope. There are 2 rings are there. First ring as well as second. One ring is present here. One ring is present within the nucleoplasm. Another ring is present in the cytoplasm. First one, this one is what? Cytoplasm. Then this one is what? Present in the nucleoplasm. There are two rings are there. One ring is present in the cytoplasm and other one is present in the nucleoplasm. At the center, the central ring is there. In between which one? Two rings. There at the center, there we want to see what? Central ring is there. Then the nucleoplasmic ring, that ring is called what? Nucleoplasmic, the nucleoplasmic ring as well as cytoplasmic ring as well as central ring, all together. There are three rings are there, okay? Which one is what? First one is nucleoplasmic ring, cytoplasmic ring, then central ring. All together, all these three rings together, they will call what? A scaffold. Which one? Cytoplasmic ring, nucleoplasmic ring as well as central ring. Together, together they will form what? We can call them as what? Scaffold. Then from the nucleoplasmic ring, then the next one, nucleoplasm. The first one is what? Nuclear envelope is there. Then the second one is what? Nucleoplasm. Within the nuclear envelope, we will see that matrix is there. That matrix is from what? Nucleoplasm. The nucleus contains a semi-fluid matrix that is called as a nucleoplasm. You can call it as a karyoplasm or it may be karyolymp is there. Okay. So within the, as we will see within the cytoplasm, there are some nutrients are there. As well as nucleoplasm also consists of nucleic acid. That one is what? DNA or it may be RNA is there. DNA is there as well as RNA is there. Then the proteins, enzymes, then the minerals, vitamin, sodium is there, calcium is there, magnesium is there. Okay, all these nutrients are present. Then the proteins are two types. Within the nucleoplasm, there are two types of proteins are there. Histone protein, these are called as basic protein. Okay, then the non-histone protein, these are called as acidic protein. Then the histone proteins are nucleohistones as well as nucleoprotein. Once again, it will be classified. Then the non-histone protein, these are what? Phosphoproteins. Okay. Then the next one, enzymes are, this nucleoplasm, it is also consists of some enzymes are there. That one is what? DNA polymerases. That one is very important for DNA replication. Then the RNA polymerases must and should it, if the replication it will be started, must and should need what? DNA dependent RNA polymerase enzyme. You will study in the molecular biology. Then the NAD synthetase, aldolase, enolase. They also contains cofactors, adenosine triphosphate, coenzyme, acetyl coenzyme A is there. Okay, these are present in the nucleoplasm. Then the next one, chromatin. Whatever within the nucleoplasm, you will see the thread-like structures are there. These are called what? Chromatin threads. Okay, a network, network of colored threads seen in the nucleus. It is called a what? Chromatin network. Within the nucleoplasm, you will see what? Thread-like structures are there. These thread-like structures, they will call what? Chromatin network. Okay, it is chemically composed of, in the eukaryote only, not in the prokaryote. It is chemically composed of 40% of DNA is present and 60% of protein is present. Okay, then based on the degree of staining, we want to classify it into the two types of chromatins are there. First one is what? Heterochromatin. Second one is what? Euchromatin. Within this, these all the thread-like structures, these are called what? Chromatin. Then that chromatin, it will be classified into the two types. First one is what? Heterochromatin. Then the euchromatin. Okay, have you got it? The first one is heterochromatin. Okay. These are darkly, here some regions are there. These are what? Dark in color. Yeah, these are what? Dark in color. Okay. Then these are darkly, they will condense part of the chromatin. Sir, how they will become dark, sir? Here, this one is what? Chromatin. Once thing it will be condensed. Then it will become what? That chromatin thread, if it will condense, together we will get what? Dark region. Okay, that is called what? Heterochromatin. Then it will contain little amount of DNA and more amount of RNA. Okay, then genetically it is metabolically inactive, that heterochromatin. Then the euchromatin. Next one, euchromatin. These are what? Lightly stained. These are darkly stained structures are called what? Heterochromatin. But light stained structures, these chromatin threads, those are called what? which one? Euchromatin. Then it will contain very large amount of DNA and little amount of what? RNA is there. Then this is about the chromatin. Then the next one, nucleolus. Here you will see another dark region is there. Yeah, this one is the what? Nucleolus is there. Okay. Then it was the first time it was discovered by Mr. Fontana. Then it is darkly stained. Then it may be spherical, it may be dense, it may be granular body. It is membraneless. You must mind it. Within the cell organelle, within the cell biology, there are some membraneless organelles are there. It is also one of them. Okay. This one is what? Nucleolus. It does not have what? Membrane. You must mind it. Okay. Then I think membraneless. Then calcium ion. See calcium ion. They will maintain the shape of the nucleus. Within that nucleus, you will see. I think calcium ions are there. They will which one? They will maintain the shape of the nucleus. Then I think this one. It will consist of three regions. First one, within the nucleolus, we will see the three regions are there. First one is granular region. Then I think containing ribonucleoprotein. Then second one is fibrillar region containing I think fibrils of the proteins. Then the last one amorphous region. Then it is composed of, along with I think within this nucleus. We will see rRNA, ribosomal RNA is there, proteins are there, some enzymes are there. Then it is also associated with the nucleolar organizer region of the chromosome. You must know, okay, mind it. Then this nucleus, it is another fantastic, it having another fantastic character. It will disappear in the, I think, prophase phase in the mitosis. Okay. Then it will once again reappear in the which one? Telophase. I will teach in the next class, I think, cell division as well as cell cycle. Okay. It will present in the prophase. It will disappear in the it is disappear in the prophase and reappear in the telophase. Then what is the functions? Already you will know. I think this is the structure of nucleus. I think it is helpful for the transmission of genetic information from one generation to another generation. What I am today, I will get the information from my parent through the DNA. Okay. Then that DNA, it will present within my father, within my mother. That will comes within my body. That is what it will that nucleus. It will help for the transformation of DNA or the genes or the genetic material from parents to offspring or one generation to another generation. Then it will control all the metabolic activity of that cell. This is complete what? This is one is what? Complete cell. It will control all the activity of that cell. That is the main function of the nucleus. It will control the cell replication also. Then it will maintain the structure as well as function of the cell by the protein synthesis. Then nucleolus, it is the site of RNA synthesis as well as ribosome synthesis. Then this is the explanation of the nucleus. Then the next one, I think we want to study about the which one? Lysosomes are there. Sir, what do you mean by lysosomes? Yeah, then this lysosome, I think the lysosomes, these are first discovered by Mr. Christian Dew. Christian Dew, he is the first person to discover the, I think, lysosome. Then these are the small vesicles, small particles. Those are present within the cell. Then in the cell containing, they will containing lysosome. They will containing acid hydrolysis or hydrolytic enzymes. This is the main concept of the lysosome enzyme. They will consist of what? Hydrolytic enzymes are there. Then these are mainly found in the eukaryotes. I think 1 minute. Then these mainly found only in the eukaryotes, but not these are present in present in the RBC. That is, these are absent in the RBC, red blood corpuscle as well as prokaryote. Then the structure, it is single membrane bound cell vesicle is there. It is what? Not double membrane, as you will see in the nucleus. That one is what? Double membrane structure. But lysosome is what? Single membrane structure. It will consist of 24 different types of enzymes are there. How much? 24 different types of enzymes are there. Like which one? Acid ribonucleases, acid phosphatases, then acid phospholipases. These are enzymes which are present within the lysosome. Then these enzymes, these are synthesized by the cell. These enzymes are present within the lysosome. These are synthesized by the cell and these are packed by the Golgi complex. That is the main function of the Golgi complex. Golgi complex, it will pack it, I think. Which one? Lysosomes are packed by Golgi complex. The liposome, I think, membrane has certain stabilizing materials are there. That I think liposomal membrane or the lysosome membrane. It will consist of what? Cortisol, vitamin E, cholesterol, and they will give the stability. Okay. Then the next one is function of lysosome enzymes. Then these are helpful for the intracellular digestion. These are helpful for what? Intracellular digestion. Then whatever, I think within our body we have the reserve food material is there. It will be digested by the lysosome enzymes. Then when the enzyme they will pure in the cell. And the enzymes they will pure in the cell. The cell itself will be digested. That is called autolysis is there. Cell, it will engulfed by itself. That is called what? Autolysis. Okay. Then hence we can put what? Suicide bag of the cell. We can put what? Suicide bag of the cell. Then the lysosome, these are also helpful for what? Removal of useless mitochondria, removal of useless RBC, removal of useless bacteria. Okay, I think those are removed by the lysosomes. Then I think malfunctioning or abnormality of the lysosome, they will cause very serious diseases within our body. I think that is called as cancer. If it will do the autolysis, then definitely I think normal cell, if they will do they can undergo autolysis, then it will cause what? Cancer. Okay, this is about the lysosome. Then the next one, vacuole is there, and the cell organelle. That one is called what? Vacuoles. Okay. Then within this cell, here you will see the nucleus is there, and then here some some regions. These are empty-like structures are there. These empty-like structures, those are called what? Vacuoles. Okay. Then these are membrane bound. These are membrane bound clear regions. These are filled with the watery fluid more distantly present in the plant cell. These are mainly found in the plant cell only, but not in the animal cell. Here I want to draw one vacuole is there. This one is what? And vacuole. This one is what? And vacuole. It will fill with what? Watery fluid is there. It will fill with what? Watery fluid. Then in the plant cell, these are surrounded by what? Semi-permeable membrane is there. That is called as chloroplast. I think this one is what? Structure of vacuole. It having a membrane. It having a what? Membrane. I think that membrane will behave like what? Semi-permeable membrane. Then that membrane is called what? Tonoplast. Which one? Tonoplast. It will be asked in the in the KPSC examination as well as in the UPSC examination. Okay. Then the fluid present in that vacuole is called cell sap. Then this is composed of water within this vacuole. Within this water fluid material, we want to see the water is there, sugar is there, ions, pigments, alkaloids, these are present. Then sometimes these also contain hydrolytic enzymes are there. Then in the animal cell, if the vacuoles are present, those are very, very smaller inside. But in the plant cell, vacuoles are larger inside. Normally, in the animal cell, vacuoles are absent. If these are present in the animal cell, these are very smaller inside. Okay. Then we can call them as a provacuole. Okay. Then this is about the structure of vacuole. Then the function. These are helpful for what? Turgidity of the cell. Rigidity of the turgidity of the cell. First function. Then the next one. These are helpful in the contraction of the cytoplasm is there. Contraction of what? Cytoplasm. It will form the internal contractile machinery of the cell. It is helpful for the support as well as maintenance of the shape of the cell. This is about the structure of vacuole. Then I think next class I will teach next and the cell organelle. Thank you.